Welcome, everybody. Thank you for coming to Grand Rounds, both here in Oberdorf Auditorium as well as joining us on Zoom today. It is my great pleasure to introduce Dr. Thomas Wallach, who is Fellowship Director and Chief of Pediatric Gastroenterology at SUNY Downstate. He's as well as the Research Director for Pediatrics. He's a physician scientist focused on health equity, environment, and intestinal epithelial homeostasis, combination which will make a little more sense after his talk today. He completed his residency in Columbia and New York, and he completed his fellowship here at UCSF in Pediatric GI under the mentorship of Mel Heyman, Emily Perito, and Zev Gartner. Dr. Wallach discovered his passion for alleviating environmental enteropathy, or EED, while seeking out opportunities to work in global health and health equity on a very large scale. His presentation today will talk about how cutting-edge tools are unraveling complex biology underlying EED and stunting. Personally, I think I found working with Tom to be an absolute joy over the 4 years that he was here at UCSF, and in particular, his dedication to improving care and accessibility of cutting edge technology across domains, across international borders, with a true passion. And so I'm very pleased to welcome Tom to talk with you today about his research.
So Jim left himself out of the list of mentors that were integral to my career and my experience and the things that allow me to do this work. So I want to add him onto there. But today I'm going to be talking about environmental enteric dysfunction, which is a condition that not a lot of people talk about, and how it leads to global stunting around the world, and how we have done some work here and in combination with some epidemiology work that has advanced our understanding of it to provide some possible therapeutic options, including some ongoing trials that we have right now. So here are my disclosures. None of these are relevant to EED or anything I am talking about in this talk. Okay, I'll try to use the microphone a little more. Okay, all right, let's see. So objectives today, we're going to talk about environmental enteric dysfunction. We're going to talk about what single cell sequencing is. You hear a lot about it, but we don't usually talk about it when we present the data. So I'm going to try to explain to you why these interesting colors show what I say they're showing. And then we're going to integrate all those omic findings with other existing literature and possible epidemiologic work to talk about how you kind of follow this all up. And so before I get started talking about EED, I think it's useful to kind of think about the framework of how I got into it, which was not that I had a PhD or knew how to do science beforehand, but I recognized that there was something interesting. And I came to a place that allowed me to explore interesting things for an exorbitantly large amount of time. And so I was able to go through different things and discover new things. And in the process of developing that skill set, find opportunities to do this kind of work. And what was really interesting about this kind of work is that I didn't get a grant to pay for this. This was all paid for by finding other people whose interests aligned with mine and offering them opportunities to do something they wanted to do, which also was something I wanted to do. And especially in the current environment, that is an interesting skill to have, and one that will probably help you continue to get work done as you seek ways to finish up things if you had grant interruptions or other challenges. So EED, or environmental enteropathy, we use the names interchangeably, is a condition that is wildly common, is all over the world, is probably the largest cause of stunting in the world. And it is something that is generally subclinical, not seen in terms of symptoms, and just results mainly in the observable phenomenon in stunting. So what you see when you look at this, if you actually go through and look at their intestine, is increased intestinal permeability, decreased barrier function, malabsorption, and then stunted growth is the big outcome. But there's some secondary things that you see as outcomes as well, like increased risk of metabolic syndrome at a lower BMI. And then you also see things like decreased response to oral vaccination and other things that suggest immune tolerance signals. What you find, we generally thought that this was due to a combination of malnutrition and recurrent enteric exposure. But in the last few years, for the most part, it had shifted towards enteric exposure because efforts to provide supplementation with micronutrients didn't really do much. So you gave people vitamin A, you didn't get very far. You gave people zinc, you didn't get very far. And so people were like, okay, maybe this is really just the infections that's doing this, and it's some kind of adaptive response that's causing this issue. What we see when we look at this on biopsy is sprue. And if you see anything, you might just see a whole bunch of immune cells in the intestinal tissue. You might see sprue. But if you see anything, it's going to be that. And so nothing that's that dramatic, and generally what we would call a subclinical enteropathy. It is very highly spread, but you might notice something interesting. The red is concentrated in specific regions of the world, but it doesn't exactly align with economics. So if you look at Central America, for instance, it's not as bad. But Central America is just as poor as much of sub-Saharan Africa. South America has more money, but Central America is not. And so you're wondering, why is it so bad? And then you look at the subcontinent, which is not as poor as sub-Saharan Africa. So why is India and Pakistan having such a hard time with this if it's purely from enteric exposure and poor access to sanitation and water? So even just looking at these older figures about, and this is about 6 years old, 7 years old at this point, we can see that there's some discrepancy in the patterns of how this shows up that suggests that there's maybe something under there we should look at. So we have this condition that we think is caused by poor access to clean water, poor access to like hygiene tools and sanitation. And we're trying to figure out how to study this. And one of the problems is this is subclinical. So you're looking at this a problem in kids, and it's not something that comes with a clinical presentation. So there's already some limitations on how you can go about looking at that. And you're also looking at something that requires, for the most part, an anthropometric measurement. An anthropometric measurement literally requires us to watch children grow, which means that you have a slight time horizon issue when you're looking at these studies, because you do an intervention and you're going to have to wait a while. And that means you have to wait for the recruitment and the end. So there's basically at least a year generally, sometimes 6 months, between when you can finish enrollment and when you can finish the study. And so that barrier creates some problems. One of the ways they got around this was trying to do an 8-country study called MAL-ED. They did do that. It worked out reasonably well. They were able to identify some biomarkers. And the biomarkers you might recognize, they're really, if you know a lot about intestinal immunology, so maybe people might not recognize that, but myheloperoxidase, neopterin are inflammatory byproducts from immune cell activity. And alpha-1 antitrypsin, if you were ever remember on GI rounds, is what we ask you to look as a proxy for barrier function, because it is not that eaten that much as it comes out of the intestine and stool. And so it ends up coming out of your body and you can detect it, as opposed to other proteins which your body eats or the bacteria in your intestine eat. So we have this biomarker, and you can use other things like very intensive efforts like lactose mannitol ratio to look at barrier function as an assessment of it, but none of them work that great. So again, we're still trying to figure out ways to get better at doing a study on this and to look at this. And part of doing that requires you to understand the mechanisms driving this. You cannot find a biomarker if you don't know what's happening. And so until we do that, we can have a little trouble. So one of the ways I thought about how we could approach this, and which, again, the mentorship team here at UCSF was just like, sure, which was pretty cool because it is a big project, is to use sequencing to look at this. So one of the things I like to do when I talk about this is go through what sequencing is, because we have a lot of reports about sequencing, but I think there is, even inside of the field of people who use it to do science, some misapprehensions of what it's actually telling you. So that, in an overview, what I want to say first is that I'm going to have a lot of these figures. And honestly, I don't want you to memorize any of them. They are references. Imagine them like a citation. They are there to justify the argument I will make. But in general, even the diagrams of how we do sequencing can be a little complicated. So I don't want anyone to be concerned if they see a big UMAP or something else like that. That is like, what does that even mean? I will talk about it. But what you do with sequencing is you take tissue or another source of biological material, and then there's a couple different options you can use. You can either just like blend it all up and try to isolate all the RNA out of it, or you can try to isolate single cells out of it using a couple different mechanisms, and then isolate the RNA from the single cells. The value of that is you can find what the tissue is trying to do. So RNA sequencing expresses what is the cell trying to do at the moment that you got it and tried to get it. It doesn't always tell you exactly how that works. It doesn't tell you what the cell is actually currently doing. And it doesn't tell you anything about how it's interacting together, but you can, using a lot of metadata, try to figure that out, as well as other people's published data. So what you do is you take the RNA, you isolate it out of the cells or whatever tissue you're looking at, and you create cDNA by essentially putting more base pairs on top of it so that it stabilizes. And then you can run it through a sequencer and figure out exactly the order of what you're seeing, and then map that to genes that we know about. So this allows us to identify other issues, other problems, and see essentially relative expression of genes. So it doesn't give you a real number like quantitative PCR does. It gives you a relative expression of genes. So you can see sort of how does this compare if I look at two samples. So I can say that I'm looking at my samples and Mel's samples, and we can see the difference between us from a biological point at that framework. The best analogy I've come up for this, for those who aren't going to actually do it, is the bar game where you look at pictures and see what's different. And the thing is, what you're actually looking for is what's different. You're looking for those things, and that's how you win the game, because a lot of the signatures are going to be overlapping. Our bodies require certain things to occur to function. And so there's a lot of similar signals that are happening. And what you're looking for is what's called differential expression. What is different about those two sets? So some people did do some fecal work looking at this. So we have a nice output for looking at intestines because people poop, and poop contains a lot of shed cells and it contains immune cells. So you can get some decent ideas as a reflection of what's going on. And you can also use that to look at microbial populations. So if you look at some of the work done in Africa by one of my collaborators, you do see some interesting signals. And basically the long and short of it, if you don't want me to go through all of the various acronyms and things in the genes, is that you see essentially some signals that reflect increased immune activity, and you see signals that reflect decreased differentiation of certain cells, in particular goblet cells. And that's what you get with the decreased mucins. These decreased mucins in particular are reflective of the fact that we don't see goblet cells. And we know that about the ED because we can see that in histology, that there's a paucity of goblet cells. So what we can see is that sequencing is reflecting a lot of the changes that we're finding. We also see is some things that were suggestive of, maybe a non-proliferative environment, because that's one of the things that can alter goblet cell differentiation and other changes like that. So then we looked at some bulk tissue, or they looked at before I got involved. So this is my partner, Paul Kelly. He's a British guy who moved to Zambia about 40 years ago and is now a Zambian national. And he is one of the guys who's taught every gastroenterologist in Zambia. He created 15 of them at this point. So he's been behind or associated with every scope ever done in the country. And so he's got a lot of patients that he can pull into this. So we're looking at this and we see, okay, what are the major transcripts that are differential from the duodenum? And we do look at the duodenum because we're mostly worried in this about not a fecal output or change in constipation or diarrhea. We're worried about growth. And so we think the duodenum is one of the better options for that. Also because the jejunum is hard to get to if you're going to try to do that as a research study. Although we actually did a little bit of that too, although not discussed here. What they see is really similar to what you got in the stool. You see activated inflammatory transcripts, and you see signals that are consistent with a decrease in specialization into certain secretory cells, particularly goblet. And you see a decrease in basically some of the proliferative signals as well. So you're seeing a kind of confusing picture, because usually with inflammation, you see proliferation, because this way our tissue responds to being inflamed is trying to regrow it. You get things like epithelial to mesenchymal transition. You get a lot of proliferative effect as it tries to replace the damaged tissue or whatever else has happened. And for here, those have been decoupled. You're not seeing any relationship between those things, and it's sort of going in an inverse way than you would expect. So what's the difference between bulk and single cell? So with bulk, what we did was we took a tissue sample and we threw it in a blender, basically, not an actual blender, but we dissociated it all, and we don't know what cell it's coming from. Now, the issue there, right, is that if you look around any single room, the average person you're going to find is not the one who made the decisions about the policy for the entire organization, right? So if you go around here and you pull any random person on the floor of the first floor of this building, the odds that they are the chair of pediatrics is relatively low. And so you do not get good reflection of the cells that are generally driving this behavior. So you see, essentially, you get a reflection of the most common cells because you are actually taking the common denominator. And because there are some limits to how deep we can go into sequencing when we're looking at it at any individual sample, it's something called read depth, which you don't need to worry about. It means that you get some barriers to finding out what, for example, enteroendocrine cells are doing, or what, for example, we are seeing in relatively low prevalence cells like tough cells. And we think those are probably important to this disease. So one of the arguments, and you also can't pick out the immune compartment very easily either. So one of the arguments we said, okay, well, let's look at this for a single cell. But one of the problems with single cell is you have to get tissue into single cells and then figure out which cells they are. And that is not a trivial problem in many ways. So what we did historically to do that was we used very expensive machines that use either water pressure or like more or less electrical fields to flick an individual cell into a chamber. So you could separate out a single one at a time. Those machines are very expensive. They require a lot of calibration. They require a lot of maintenance. They don't work very well if you throw them on a plane and take them to Lusaka. And so you have a hard time doing that. And at this point in time, we didn't know what happened if we froze tissue or did something else. And if you really, to preserve it, you want to break it, the RNA and the nucleic acids out of the cells. That's the best way to ship it. But if you do that without isolating for single cells, you can't do that again later. So we wanted to figure out, how do we do this? And how do we do this in a global health context? Because that is one of the big issues that people face. They couldn't do this before because they couldn't do single cell isolation. So that was the hurdle that we were hoping to overcome. And the way I overcame that, and again, by speaking to someone else's interest, is at the same time, a lab in Boston at MIT, run by Alex Shalik, had come up with a technique called sequel. Sequel was really cool. It uses a mathematical concept called Poisson loading, to basically say, if we break up all the cell tissue into what should be more or less single cells by dissociating it and breaking all the connective stuff, and then we put it onto this tray, and then we tilt it at certain rates, statistically, a certain fraction of the wells will fill up with one cell. And you have ways to back check that and validate it to make sure you didn't get a doublet or a triplet. So you can do that, and it worked. So these guys invented this thing that we could take with us that cost a couple $1,000s instead of millions. And we took it with us, and we took the $5,000 plasma sealer, which was the only equipment piece we needed to add to it. And we all went to Lusaka, and we did the dissociation right there to make sure we had high resolution data for the study that we were doing afterwards. And what was good about that is that from Alex's perspective, he has this new technique he wants to show the value of. And the use case that he was really highlighting was being able to do single cell dissociation anywhere. So he was really excited to find people who had tissue to do single cell dissociation in a context like Lusaka, because that's what he wanted to show. And so for him, it was a no-brainer. I offered to get him tissue that he couldn't get on his own without years of work. And then he has it, and now he can be involved in publishing that paper and highlight the utility not only of this work, which he also enjoyed doing, but of his own work. And so that allows for a collaboration where everybody benefited, and allowed me to do a project that should have cost roughly $500,000 for $0 new dollars, which was great because I don't know who else would have paid for that. So this is kind of just a diagram of what happens with SeqWell. So you take the complex tissue, you break it up into those cells, you load a micro well, which is designed to have like super tiny wells, and then you basically have you use just an oscillator at a certain frequency to generate the Poisson distribution, and it works, which is extremely cool, and another triumph of math over common sense, which is most of statistics. So then you were able to do the same thing. What's inside each well is a barcoded bead. That barcoded bead tells you what you're looking at when you're looking at the sequencing later. So the barcoded bead doesn't know what kind of cell it is. You figure that out from the sequencing. But the barcoded bead tells you all of these sequences are from this well. And that lets you say this is. And you can tell looking at that if you accidentally got 2 or 3 cells in the well, and you can exclude those data points. So we went to Zambia, where there was an ongoing Gates-funded project that was getting these endoscopies. So again, we were able to do endoscopies by using an ongoing project. And what we did was Alex and I offered to Paul Kelly that we would do the single cell sequencing, and he doesn't have to do anything else. So all he had to do to make that occur was to update his IRB. That was in fact a process, but we still got through it, and we were able to go ahead and do it. So we went, and then Paul and I completed the scopes. We pulled out tissue, we did the dissociation, and we were able to take that tissue back in the form of cDNA libraries. The nice part about shipping things back in cDNA libraries is that that is considered chemically inert and doesn't have a biohazard. So it just went back with a postdoc in a backpack, which was great, and then avoids a lot of headaches about sometimes trying to bring in things like fecal material, blood, other stuff like that from Africa. So tissue dissociation on site and single cell isolation on site saved us a lot of money, a lot of time, and preserved our confidence that we're looking at shifts that were not from being frozen or something else that occurred, but from what was ever happening to the tissue at the time that we got it, because we took it from the biopsy to dissociation within like half an hour. So we were able to get 12 participants. Unfortunately, in the course of finding those 12 participants, 3 were diagnosed with HIV. So we did not recruit people who had known HIV, but part of the workup for this trial was diagnosis. So we did end up with 9 patients who were HIV negative and 3 who were HIV positive, as well as another one who I don't mention here, who had HTLV, because we weren't entirely sure how to interpret it with only 1 data point. So we had 3 regions of the small intestine. So this one, we did actually do some of the cases. We did jejunoscopy using just a push method. I did not do that because I could tell that my former fellowship directors might have some concern about my technical skill in that, which I don't have. So what we were able to find is these biopsies. We were able to obtain them, and we did the typical things looking at them under immunohistochemistry. These are actually samples from the study. This is just an example of moderate environmental enteric dysfunction or environmental apathy. This is severe. As you can see, the sprue gets a lot worse, but even in the moderate one, there's some disorganization. It's not exactly normal, and you can see a lot of immune cells. Our comparison group, because I mentioned before, sequencing is always going to be looking at those 2 pictures and comparing it, is from EOE and from patients who had a Whipple and had intestinal resection. And again, these are all adults. So this was adult patients who were undergoing endoscopy because, again, it's easier to study adults, and environmental enteric dysfunction does persist in that population. And then, so we used adult patients as a comparison. And again, This is primarily for financial reasons. It's not the perfect comparison. The perfect comparison would be asymptomatic adults that we scoped for no reason as part of a study. But this is available. It is tissue that is available that should not have a duodenal involvement, we think. But at least by using two different sources, we can triangulate and see if there's something going on. So I include this not so that you look at it. I include this so that you know that every time you open a sequencing paper, unless you do this for a living, it is a common experience to look at this and be like, I don't know what that means. And that is because most of it is not really there for that to communicate information. It's to validate what you're communicating in other ways, typically words, in many ways, unless you are somebody who does this for a living. So the biggest thing that you want to take away from this is, again, you're looking to see if those pictures are the same. And this UMAP up here is actually the best representation of that. The different colors are different samples. And if they overlap, they're the same. And if they are physically different looking, then they're not. That is basically, it's reductive, but that is more or less kind of what you're looking at. And everything else is a report of differential expression. So it's all just how much more or less are we seeing this expressed in the two groups that you're comparing. And you have to remember, everything is relative in a sequencing comparison. It is a different thing to look for quantitative assessment of RNA. So because this is complicated and confusing, I'm going to start with the end. So I'm going to tell you what our conclusions were, and then I'm going to explain to you why the sequencing links up to it. So what we found after all this was essentially what's called we're calling a dormant defense phenotype. We view it as a little bit of an adaptive response to recurrent enteric infection to essentially mitigate some of the inflammatory changes and days of diarrhea so that you don't die of dehydration or something else like that. But it also causes a bunch of secondary changes, and it causes some major immune regulatory and immune suppression effects. So what we see is epithelial remodeling and dedifferentiation. Dedifferentiation refers to when a differentiated cell type like a goblet cell or an EEC decides it's going to become epithelial again, which had been shown in theory in animals, but had not been shown in humans. We were the first group to find that. But it allows you essentially to have a different direction of it, which creates sprue to some extent, because as you dedifferentiate, you physically move down the villus again, which is weird. And people don't think about the fact that our villi are basically escalators, but it literally essentially reorders the villus to make it shorter as you do that. In addition, they will reshape themselves into something called surface mucosal cells, especially in the bulb. That's maybe an H. pylori indicated thing, but what it is these are essentially non-absorptive enterocytes. So it's almost like when you get in the esophagus, a Barrett's esophagus, to be able to protect you from enteric infection and probably from stomach acid, it remodels itself to be a non-absorptive enterocyte. So it becomes flatter and less capable of absorbing and doesn't work as well. But it does do pretty well at existing as a barrier still. And that's better than if you're just letting your intestine get blown up all the time by infection. And then what, again, I see is a disconnect between inflammation and proliferation. Because if you look at IBD, for example, both things are happening at the same time. You're getting inflammation and it's making your intestine try to make more of itself to repair it and heal it. And we don't see that. So we see mild inflammation and then no proliferation, which is confusing and suggests something else is going on. What we also saw was a really, really strong signal for tryptophan deficiency and other amino acids in particular, but basically an animal protein deficiency because they weren't eating animal protein. And we saw a bunch of sequences that are associated with that and have previously been associated with that in other people's work. So the first step we did, whenever you're doing sequencing, you want to make sure what you're doing is reflective of the tissue that you're looking at. So it's called validation. So we know that in EED, people lose their goblet cells. So we looked, did you have the goblet cells lost? And yes, they did. And we said, okay, we see something called best 4 enterocytes. That's a marker that appears in certain enterocytes. Did we see those as well? And we did. So our data is aligning at that point with previously published data and known histology. So it tells us that we didn't mess something up from the go. The other thing that we can look at is some of the immune compartment and say, hey, what are we seeing there? And we're seeing the same stuff that people have seen in previous sequencing and stuff that's consistent with histology, which is a highly activated immune cell compartment that's acting like it's angry. And that is, again, consistent. So what that tells us is we can keep going. We have not hit the first roadblock and created a giant problem for ourselves. So the prior studies that looked at this had found a couple of different things. And this is, again, those surface mucosal cells I was talking about. So this is something called DUOX2 is the major expression factor here. And people had previously noticed this. And so we said, oh, this is weird. Let's take a look at that. And they were extremely common. They're generally in the stomach, which is probably not too surprising because they protect from acid to some extent. But we also found them in the duodenum, and it was highly associated with the severity of EED. Now, it was also highly associated, when we look later, and I'll get to that, with H. pylori. So the question is H. pylori contributing to the severity of EED, or is this from H. pylori, or is it all the above? And my theory is all the above. When we look at the bulb here, we do see, again, that this is being driven by dedifferentiation. So you're not making surface mucosal cells, they become surface mucosal cells. So it pretty much, your enterocytes are Other cells see this stimulus, which is either recurrent infection or H. pylori-induced acid, and say, hey, I don't like this, and they go down. And it's quite possible, and I've seen some literature suggesting this is how peptic duodenitis works, which is what happens when the stomach, in the duodenal bulb, when you have a lot of gastric acid, it causes some changes to the way the bulb and the duodenum work, and this might be somewhat similar. So, and again, as you notice, I'm mostly having the figures as a reference point. So I'm not going to talk too much about individual figure, but you guys have the slides if you want to check it out later. So this is looking at dedifferentiation, and what you're seeing, essentially, you get a migration of cells back the wrong direction. So you're reversing the direction of the escalator. You're supposed to start in the crypt and go up. And in dedifferentiation, they more or less pull out the cells from the top and they start to redirect where they are. In some extent, it's just from flattening and other physical changes and morphology changes. But to some extent, there are shifts in actual directionality of cell movement in the LG. Like when you look at what's supposed to happen is you start at the bottom and the stem cells differentiate and move up, and you can track individual cells. And they start not doing that in part because of the also cessation of proliferation. So because there's no replacement cells driving up the escalator, sometimes they stop working right and it goes back down. And I see Jim looking at me like, hey, that's not exactly how it works. These are very reductive metaphors. So just to be clear, these are reductive metaphors, not specific things. This is a way to understand the data that I'm presenting. So relative to US samples, the environmental enteropathy patients had a whole bunch more enterocytes and lower abundance of plasma cells. This is essentially consistent with the anti-proliferative effect. You lose a lot of the gradients that drive differentiation of stem cells into other cells if you don't have a proliferative effect. And because a lot of that is crosstalk between the cells themselves. And so what you end up left with is basically just structural cells like enterocytes, and a lot of them have become SMCs and they don't absorb food very well. So this allows you to have some of the dysregulation that we see, where you see like minor GI issues for folks and you see metabolic issues, because as we all know in the modern era, the intestine makes a really important set of chemicals that control our metabolism. And so if you lose a lot of the cells that do that, there's a pretty good chance you could also impact things like GLP-1 production and other things like that that are heavily involved in metabolic regulation. That might have been foreshadowing. So the other thing was just revisiting that we see all this inflammatory signaling. So we see some of the inflammatory signaling that overlaps with IBD, but it's a disparate presentation. So we see some inflammation, but we don't see a lot of the secondary signaling that goes with IBD. And this is, again, and I keep hammering this because this is the hook, it doesn't have proliferation, and that's weird. And this is something that we see in some other conditions. Like you see a little bit of this sometimes in people with Hirschsprung's and Hirschsprung's enterocolitis, and you can see other factors of this. But it's strange to have that effect separated from each other. So this is just, again, just supporting that with more data that you can refer to later. So the decreased proliferation was heavily associated with tryptophan. You might be like, ooh, why is tryptophan associated with that? Well, tryptophan is a precursor of kynurenine, which is a precursor of serotonin. And some really great work out of a woman named Kara Margolis, who's a PhD now at NYU, demonstrated about 10 years ago how important serotonin is to trophic growth in the intestine. So serotonin, particularly from the enteric nervous system, drives the proliferation of stem cells. You get rid of that serotonin, and you do not have nearly as much proliferative signal. Serotonin in the intestine itself from EC cells is a little bit more pro-inflammatory. Getting rid of that, and you might not have an epithelial response to inflammation, even if you have a systemic inflammatory signal. So to me, that suggests a little bit of something suspicious, that maybe tryptophan is involved in this and very relevant to the process. So we also had a couple other cool things. We had changes in ILC3 and gamma delta T cell fractional abundances, which reflected IL-22. One of the things you can tell, even if a sequence is not common enough to show up in sequencing data, because again, even if you're getting the cells that are in charge, some of the things that actually drive cell behavior to change it are actually still really rarely expressed. So you could have a 6-fold increase, but if you only make 2 copies of that gene, 6-fold increase is not that detectable. So you can impute from other things. So IL-22 is one of those relatively low production ones. By looking at some of the downstream effects and some of the cell differentiation that we see, we can impute a decreased IL-22 signal in environmental enteropathy. And that is also interesting because IL-22 is involved in Crohn's disease very heavily. So we are seeing this picture that's a little interesting. So this is, again, I've mentioned this before, and probably noting that I repeat stuff a lot in this talk, and it's because sequencing is, again, very dense. But IL-22 is known to be involved in Crohn's disease in particular. And what it drives is hyperproliferation, endoplasmic reticulum stress, and it uses the ATF3 signaling axis. This is one of the genes that's commonly linked to IBD, in particular Crohn's. And it is a pretty common expectation. We see increased cell death, apoptosis, necroptosis, whatever else you're looking at. But to replace that, you see a proliferative drive. Now, in our picture, we see malnutrition blocking that. So what we're proposing from our sequencing data is that the tryptophan particularly, but really essential amino acids, but tryptophan is the biggest one, are not present in a lot of diets in these areas. And if you remember that map, one of the things that's not commonly eaten in India is animal protein. And so it could explain why, for example, India, which is the richest country to have that degree of stunting, has that degree of stunting, especially because they still have that degree of stunting. In the last 20 years, they have done insane amount of modernization of their hygiene infrastructure. So there is a ridiculous difference that's not being reflected by clinical outcomes. So the other thing that kind of hooks together here is we know that these people are still more susceptible to infection. Part of that is the immune tolerance effect, but part of that is like you need a barrier. That's the wall, mucus, and you don't have goblet cells. So now you have all these bacteria that can just walk right up to the wall, and there's not even a checkpoint anymore because it's all falling apart. So what's supposed to be a brick wall is now a chain link fence with a bunch of holes cut through it. And what's supposed to be a little like divider strip in front of it is now gone because you don't have a mucus layer either. So you end up with a lot more interaction of your immune system with this. It's part of why we think it is somewhat adaptive to suppress that. Because if you suppress that, you don't have constant disease, constant days of disease, even if relatively minor. So there is an evolutionary benefit likely to suppressing the epithelial inflammatory response, even though you are still getting that blood response from stuff leaking through and causing your immune system to get mad. And that's also reflected by this. We see a lot of activated T cells. So a lot of mad immune cells, but they're just hanging out. They seem to be totally disconnected. Usually when they send all these signals out, the epithelium around them is like, hey, I see you. I'm also going to get mad. And then it actually self-amplifies and you see an amplifying effect. This is how the whole principle of TNF alpha inhibition basically stops disease. But you don't see that. You just see these activated, hyperactive T cells, and you see a shift towards activated phenotypes. This is hard to see up here, but basically what we're just reporting when you see these violin plots is sort of where the density of cells are. And so again, we see a shift towards activated T cell phenotypes in these patients' immune compartment, as well as activation of genes that we would say are linked with that. And so this was basically when we get to this point, we're looking at validation. So this paper took 3 years to get from when we first submitted to publication. And why did that happen? Well, it's because we submitted it to fancy journals. And fancy journals ask you for additional studies. So what they asked for us was some validation of what we found. They didn't like my initial theory of validation that we found this and that it was consistent with previously known information. They wanted us to find this and stain for it. So just so we can make sure no one is concerned that maybe we're just reflecting some sequencing magic, here's some duox. So we stained for it and did find that it was overexpressed relative to healthy tissue. And so that's a validation of our sequencing outputs, again, reflecting that we are in fact taking satellite imagery of reality and not Narnia. And we are seeing that, again, this really says that we're probably seeing the surface mucosal cells, I made the wrong acronym there, show up as a defense mechanism. This is the H. pylori data that I was talking about. When you're using single cell sequencing, you can do something called Kraken. There's a couple other approaches, but you can look at the tissue-associated microbiome, and you can find signatures of certain bacteria, and to some extent, viruses or fungus if you're looking for that as well. What we looked for here was really H. pylori. We did find a decent signature, and it was highly associated with the most severe cases. H. pylori is known to also create immune tolerance effects. It's quite possible that there could be some linkage between that and a more severe version of this. Other relevant things, we did find some other cool stuff. So one of the things you might have heard about is something called the hygiene hypothesis, which is the idea that your exposures and you're growing up are what drives a lot of your risk for allergic and autoimmune disease. So, this actually, I was early on that explanation. This one is about the GLP-1, circling back to that. So this is basically, we found a UCP2 upregulation. UCP2 decreases GLP-1 production. And so even for the enteroendocrine cells you have, you might be decreasing the natural production. Now, one thing I mentioned at the beginning is these patients clinically have metabolic disease at a lower BMI. So you develop metabolic syndrome, you develop other issues at a low BMI. Now, a lot of these patients are actually from Africa. So in my cohort, we don't see one of the main symptoms of that because there's a relative protection against MASLD, but we do see a lot of issues with people having things like type 2 diabetes and other complications of metabolic syndrome. And that is something that if you look at US cohorts, you can see in maybe what you got taught in medical school about, for example, the tendency for Asian Americans to have higher central adiposity. That is actually generational. And if you go through generations, that tendency goes away and the increased metabolic risk in second and third generation goes down. And so you're looking at probably environmental exposures and epigenetic shifts. And we have some other fun work we're doing in IBD about that right now. We also see an increased tough cell abundance. This is probably not that surprising. Tough cells respond to parasites. Parasites are endemic in these regions. And so we are probably seeing that as a reflection of that. But that does have some difficult to interpret finding. We don't know exactly what tough cells are doing because we have a good idea, a better idea now than we used to when I did this work, but they are still a little bit mysterious and hard to find. So while Dr. Bayrer is figuring out a lot of that stuff on people like him, it's still not entirely clear what they're doing in this picture. So circling back, I hope I was able to summarize this and make sure that this made sense to you and that you think, oh, like that data does in fact show those findings. So again, but you don't finish a project and say this is done. I got into basic science because I was interested in global health and environmental enteric dysfunction. When I came here and interviewed, that's why Mel gave me my backwards fellowship, is because he was like, this guy has a plan. He's got something he's excited about. And it's what I did with my time. And so I learned all these skills because I was curious about this question. And it has been a great experience and I'm really happy I did. But then you're like, okay, so I'm still curious about the question. So what we found there was, it looks like it's protein. So then the question is, okay, what do we know about that? So we have those data. What do we do? So I took a look at literature that exists. I found some literature suggesting that animal protein intake was linked to linear growth. There's some great studies out of Iceland that look at that. There are studies looking at this in the region that show that animal protein intake was correlated with severity of stunting in the global south. So what do we do to try to figure out if that's real? Well, there's a couple options. I could try to do the same study, but with single cell sequencing in kids. I could try to do a therapeutic intervention. So SSRIs, for example, increase serotonin in the intestinal epithelial microenvironment. That might drive a proliferative effect. In fact, it's known to do that for people who are on them. And then, or you could use a sertraline derivative or just tryptophan as a thing. So you could try a clinical trial. And then the other option is you could do some epidemiology and look at published data sources and see if you can find any patterns. And this was, I graduated in 2020, didn't have any grant funding at that time. So what do you think I did? So I learned some epidemiology. So I found a partner. So we looked, this is a summary of the existing data. So basically, like I said, there is data suggesting that animal protein intake in particular correlated with severity of stunting. So they're inversely correlated. And then you also can look at this and see that there are lab data that's finding this. So kynurenine serotonin ratios are very significant predictor of severity of this. Tryptophan kynurenine ratios are very heavily associated with EED severity. And serotonin is, again, the thing that we think is happening. We think tryptophan pulls us all together. And we're seeing a lot of stuff. And then in the interim, while we were publishing the first paper, my partner did a trial where he gave essential amino acids and he showed improvement. So, and that was in adults. So it limited and it was a sequencing-based improvement, but he showed improvement. So we've got a lot of arguments that were there. So I'm not saying that you need to eat meat. Vegetarianism is absolutely great. There's no problem with not eating meat. The problem is that meat is the best source of essential amino acids. So there are 9 essential amino acids. They're only together in one source of protein. Every other time, if you're eating vegetables, you're going to need 2 to 3, and you need to eat them in some fairly intense quantities if you're going to be that few vegetables. Otherwise, you need a lot of different vegetables. One of the major issues in the global south is monoculture. A lot of people eat 2 to 3 things. And so it's very difficult to get all these things from there. And you see some deficiencies, especially leucine, lysine, and tryptophan that we know are linked to growth and are probably driving some of this. So that suggests some interesting things that we could potentially look into, because trying to supplement with just amino acid is expensive. So think about what we're trying to do. We're trying to intervene on hundreds of millions of kids. So if you come up with a plan that involves using a very expensive drug, it will never happen. So your goal needs to be finding a therapeutic intervention that uses something cheap. And even amino acid formula is a little too expensive. So can we restrict this down to maybe 1 amino acid? And that's what we're trying to look for here. So here's our proposed mechanism. This is, again, just as a reference for you guys to have, is that basically you get deficient amino acid diet. So you don't make IL-22, you don't make proliferative signaling from serotonin. So you get a general barrier function deficit, you get decreased goblet cells, increased immune interaction, and as a response, an adaptive response trying to suppress localized immune response. So again, we're going to follow up on this. So we started at the most local level, at the microscopic RNA expression level, and now we're going to look at things at a higher level. So I got a partner, Diana Rosenbaum, who's an epidemiologist, and she looked through Gapminder. Gapminder is a great resource for just kind of doing this as a pilot, and you can look at that, and it will point you to the actual data sets it uses, so you can get the raw data directly from the UN or the WHO or whoever you're talking about. And you can find a whole bunch of different things in there. So we were able to find global, basically local animal protein production, so we could tell how much animal protein is available in the economy of a country. We can say GDP economic data, linear growth data, and we can find water quality data. So now we have all these variables we can play with and see what's going on. What you might see is on the left, you got animal protein. On the right, you got water quality. You might see that there are actually a few more outliers on the right. But it does seem like they're basically relatively similar in terms of their relevance. Now, there is more complicated math we can do to look at this. And this is an example of one of those. So we are looking at this in terms of association, in terms of strength of the association. This does not show any causality by any means. Nothing I'm doing today shows causality. That's apparently not the kind of work I like. But what we are showing here is that there is essentially an equivalent protein and water factor, and it's very much linked to GDP. None of that's surprising. But what's surprising is the importance of protein. And then when you look at some of the specific countries, what you might see, right, is that if you look at India is kind of a weird outlier in terms of sanitation quality, but the prevalence of stunting. And what you see is that we see less of these outliers that you would expect because it is clearly a major part of this. So we're saying, okay, let's look at this. So these on the right, you see essentially the factors, essentially how much of a factor we think something is on growth. So water quality is still the biggest thing, but protein deficiency seems to be a necessary precursor to have that happen. So protein deficiency in the United States, you don't get EED. Protein deficiency in place with dirty water, you do. And that's the big difference. It's a two-hit thing. So then we said, okay, can we look at this at a micro level? Because again, that's national level. That's not talking about anyone's actual consumption patterns. All that protein could be being sold to someone else. We don't know. It's just what they make. So let's look at a micro level. There's something called MIX, the multiple indicator cluster survey. This is something where they ask families around all over, but it varies a little bit by what they actually ask by country. And they say, okay, there's a bunch of different things they collect, and they send health workers out to collect this every few months. There were 4 countries out of all the mixed survey participants that had data that we looked for, so greater than 10 years, and they actually broke apart protein by what you ate. And then you can use certain things. There's a technique called entropy balancing. I do not understand it. Please don't ask me about it. But my epidemiologist partner, who has a PhD in this, does understand it. And what it does is it reduces some of the factors like SES when you're looking at specific variables. So we deployed that looking at this. And what we found was it was meat. Eating beans didn't help very much. Eating meat helped a fair amount. Eating dairy helped a fair amount. And that makes a little bit of sense because, again, tryptophan concentrations are much higher in the types of protein sources that produce better growth outcomes. So this, again, points towards our association-based analysis in these 2 ways triangulates and suggests that we have something. It still doesn't show causation, but it makes us feel pretty confident about it. So then we kind of look at some of the other things. So based on some of these preliminary data, people started doing some studies on this. And we were seeing, okay, like this does seem to do some things. If you look at efforts people have made to put chickens in homes in Africa, that's one of the better interventions. And it's not because, and it only works if they don't eat the chicken, which requires pretty routine visits because it's pretty hard to give someone a chicken and be like, don't eat this because chicken. But if you grow it for eggs, kids do better. And so you see some really interesting found there. So one of the things I'm interested in pursuing, and we are actually currently pursuing in a clinical trial that just started enrolling in Zambia, is tryptophan-enriched formula and see if that helps, because that is something that is a SKU that formula companies could probably make for not that much more than current formula markets, and that would get purchased. So people would use this and replace regular formula, which would be the Global South formula. Other options, SSRIs do actually increase serotonin in the microenvironment. They do cause a proliferative effect. They are extremely safe. However, there are some IRB concerns and people's perception concerns about using a therapeutic trial of even low dose SSRI in children. And what we're looking for here is something that's going to impact you before age 2. So using an SSRI is a little bit fraught, but what you could do is use a serotonin precursor or just a tryptophan supplement directly. And there are biologically available serotonin precursors you could use if you think that's there. Question is, serotonin not just do you have enough serotonin, it's where it's coming from. So I'm not entirely convinced that the precursor will do it, but Kara, who knows more about this than me, seems to think it's plausible. So we'll find out. And then what was also interesting, might be interesting to Dr. Heyman, is that we found some interesting signals suggesting an inverse presence from IBD. So we see some strange variations in signal that would essentially more or less make IBD very hard to form. So increase in dopamine 2,3 dehydrogenase, this is driven by the tryptophan deficiency. So this is basically your body, this is what makes kynurenine, and your body's trying to make a lot more kynurenine, so it upregulates it. It also has an immune suppressive effect and upregulates IL-10. So then we have interferon gamma. Interferon gamma can go either direction. It can be upregulating by IL-10 or it can upregulate inflammation. This looks like it's activating in the immune tolerant sense. Then we have decreased IL-7R, which is a key driver of memory cell activity, and then elevated IL-10, which is not surprising given everything else that we show, as well as decreased IL-22 and ATF3. So again, we're looking at this thing, the protective effect in the gut that is blocking you from having the inflammatory response to all the infection and inflammation in your blood is also very likely stopping you from developing an autoimmune response and stopping you from developing any of the other autoimmune diseases we get at the intestinal interface, which is to say celiac disease or food allergies, other things like that, because it's limiting your body's ability to see that. And so it all ends up in the blood, but you don't get the same response from things in the blood as you do from the gut. It's a different antigen presentation. So we think that this is likely reflective, and I have been trying but failing so far to get a grant to look at this a little bit more intense, because I think that we are seeing something that is heavily consistent with what we'd expect for the hygiene hypothesis, which is it's not just exposure, right? If it was just exposure, South America would look much like sub-Saharan Africa, and Southeast Asia would look very different because the hygiene infrastructure is different. It's a mix of these two hits, and it's the interaction of these two effects. And it's a way that social elements like the high prevalence of religious vegetarianism and veganism in India can drive a health externality that could be replaced without forcing people to change their behavior. So if we add tryptophan supplements in India, we might see a major benefit. So these are my acknowledgements. Thank you again to these three people and Zev Gartner, who was across the street in the Genentech Hall. Thank you to my team at Downstate, Janet Rosenbaum, Yangsheng Wang, and Sam Abariga, the team at MIT, Connor Kumalo, Alex Shalik, and Travis Hughes, and the folks, of course, in Lusaka, and all the patients who were incredible and let us do this. And to the Gates Foundation and the Broad Center, who paid for most of it, and not directly to me, because again, you can do a lot. I also want to give everybody a nice warning about the dangers of copy-paste. This was my clinical co-fellow, Naomi Patel. She's over at Oakland. She came to visit me in Brooklyn a few weeks ago, and these are our kids. And as you can probably imagine, they're not very good pediatric gastroenterologists. So that's why you don't copy and paste.
So Tom, a lot of people have looked in mouse models of dedifferentiation, mostly focusing on the secretory pathway, although absorptive pathway can do it too.
Yeah.
In your single cell data set, did you see differentiation from both secretory and absorptive cells?
Way more from secretory. It was almost entirely secretory. We got a little bit of signal from dedifferentiation of epithelial, but it was much more secretory lineages.
And the other thing that I noticed in your data, it seemed like in the EE, you had a higher proportion of OLFM4 LGR5 stem cells.
Yes.
And so is the problem here that you have more slower cycling cells?
That's what I think.
And less rapidly cycling TAs?
Yes, I think that we didn't prove that, but I do think that is exactly what's going on. So basically what was noted was that we have a lot of stem cells relative to other abundances. And that's in part because some of the things that are driving the absent proliferation are essentially an absent differentiation. So the stem cells that we do have, you're not making that many more of them, but they're just sitting there. And part of that, if you read more about intestinal differentiation, it has to do with the fact that you need populations of the other cells to generate the gradients that drive that effect. So by the time you start to create it, it can be somewhat self-sustaining. So I do think that's what's going on. We couldn't prove that from there directly. And one of the other challenges is you're still looking at essentially fractional abundances. You're looking through a mirror at a mirror. So it's not always ground truth, but that is generally what we see also in stains. You see a relatively high amount of LGR5 cells and a just paucity of nuclei, more or less, in the epithelium, and like a suggestion of just like less cells. It's just less cellular. There's bigger and there's more space between.
You have like 3 questions on.
Oh, okay.
So I may need to.
Sorry.
If anyone, I'm gonna bring them up here because they're all.
Well, I was even okay on time.
Okay, so Jan, read it. So
So Janet, who also is in the division of PGI and a fantastic nutrition researcher, asked, did you consider looking at historical data in, I'm guessing, South Africa?
SSA, I don't know. Social Security.
Social Security Association. Yeah, that's it. To see a huge reduction, because there's been a huge reduction in stunting in the United States over the last 100 years, and you can look at water and protein by changes in stunting prevalence. So this is a great idea. I have tried to do this before. One of the issues in the United States is that water quality is reported at a state level. The areas with the worst water quality do not report their water quality. You can imagine which areas those are in the country. They are also the areas with some of the highest prevalence of stunted growth. So I do think that there is there, but I can't prove a water quality association because there is essentially no data. The US previously funded better data collection on water quality around the entire rest of the world than we do outside of in our borders because everything is done state by state. So I have great data on water quality in California and in New York and in Illinois and in Maryland, but not in my areas of interest in Alabama, Georgia, Kentucky, Tennessee, where I'm looking and seeing a lot more relative rates of stunting. And so I do think that's an interesting option. We have looked at different ways to try to assess that. It's been hard to pull off. Matthew Kahn, who was fellow here when I was here, and still there, says, nice things about me, which I won't repeat. When we see villus blunting in our patients with immune deficiency dysregulation, do you think that might also be related to dedifferentiation, though caused through a different root cause? I do think there is a component of that. You are essentially, especially for oncologic issues or immune deficiency dysregulation, you are probably also creating anti-proliferation signals. And so you will see some element of dedifferentiation as a response to pathogen presentation in the intestine. I also think that the serotonergic component of this is pretty major. One of the things that we're seeing when we look at other types of enteropathy is we see serotonin depletion at the ENS. And that is a common theme that we're seeing in essentially non-inflammatory enteropathy around a lot of different disease types. So it seems like a kind of emerging theory of how the intestine breaks when it's not just neutrophils doing it. And so we're getting some interesting data there. I do think it's related, but intestinal epithelial behavior is very, very stochastic. And so it's probably a couple different things driving it. And then Rachel Rodier. So says also nice things about me, which I will internalize but not repeat. She said not sub-Saharan Africa. I will get back to that. Janet done. What advice would you give about supporting scientific progress this time on uncertain extramural funding? So I actually have a lot of things about this because this is one of the other big things I do. I think that the way we do this, especially in pediatrics, is by essentially unionizing everybody. And what I mean by that is we need to be collaborative. What we have is a circumstance where even large centers like this one generally do not have enough patients to do a single center study. To do multi-center studies, if you are not collaborative, requires a lot of money because you need to form legal agreements to exchange data. You need to pay for people to do things. You need to create infrastructure every time you do it. Now, what if we just had the infrastructure and we all agreed to share our data in national registries or other things like that? And then the other thing I really want to do, and one thing I'll be pitching to NASPGHAN, which the next president was a little interested in, is essentially creating a meta CRO at our national level. So if we have our national organizations pick up, basically, we all form agreements with the umbrella organization that represents our field, and then we send them data in a registry. They have the budget and resources to work with individual funding groups like industry that would be willing to pay for us to maintain that registry. And then when industry wants to do a pediatric trial, instead of contracting a very expensive research organization to find those sites for them at a very high rate, they contract for a very reasonable rate your national society, which tells you exactly which groups have the patients they're looking for and which groups are interested in looking for it and have the capacity to take that on. And so you've now created an easy data set that can be used by us internally to do research at a broad level. So we could look at this, and I got this idea in large part from using Trinetics. And I don't know if anyone's used that before, but it's a great large-scale data set that has real-time, slightly delayed access to all categorizable data for 170 million Americans. So it's just linked to the EMR, and it sends PHI strip data, but it's anything categorizable. So that's ICD-10, CPTs, whatever else you want. And it is a gold mine for outcomes research and a gold mine for research training. I have been using it, teaching that with our residents, how to ask the question of the data that they can get, because you can ask it almost any question. But if we do this, and that company emerged out of this idea, it was to help companies find people for clinical trials. But if we make clinical trials cheaper and easier to do here, people will come and do them. One of the big barriers to getting INDs for pediatric drugs is that it's expensive. So if we support that and we do infrastructural things to do that, it will help. The other thing I will say is one of the big barriers that emerged, and one of the things I think is valuable about physician scientists, is I cannot count how many times I was in a lab meeting across the street and they said, oh, we figured all this cool stuff with our culture cells, but we don't know how to get tissue. We got a lot of tissue. And what you need to know, and part of why I like giving this lecture, is if you know what sequencing is, even if you're never going to do it, you're never going to open up R, you're never going to do your own interpretation of your data, you know how it can be used. And I have another project that Jim heard me present about at DDW, where I just use sequencing to do something to look at it because it was a relatively cheap way to ask one question. And we were able to do it. And I didn't do most of that analysis myself. I have a partner did that. And you don't need to do it yourself. You can get a partner to do it. But if you know you have tissue and you go to someone who has the machines and the expertise, they might be willing to do that because you can honestly often throw tissue on to another run you have planned and it costs no extra money really. So you find that there are opportunities to get really interesting high intensity pilot data with people who have existing funding in other ways. And so there will still be people with industry funding or foundation funding who are doing work, and you will find ways to multitask with them. But the way you do that is by showing them how it benefits them too. Because it's not that not that they wouldn't want to help you, but you have limited resources. So you have to apply your resources and your grant funding in an honorable way, reflecting your priorities, what you got funded for. So if you are not reflecting someone's interest there, it's harder to get for them to be allowed to use it, even because you have to justify to your sponsors what you did. So you want to find ways to make your work interact with someone else's, especially when they're already doing it. So this was a case where I did that with two different groups. I found a group doing a study in Africa that could get tissue, and I found a group who had the sequencing. And you might ask, what was I there for? Well, I made an introduction. And honestly, that was the most important thing I did. I did a lot of this other work, but like the biggest thing I did was say, hey, you guys should do this. But to do that, you have to know what's available, what it is, know about the problem. And physicians come in with a holistic knowledge that basic scientists often don't have because they get very focused into their area. So it didn't occur to the people across the street in my lab that this is a women's and children's hospital and we do breast reductions and cancer resections all the time, and that their research on formation of ductal breast tissue could be very heavily supported by finding a surgical colleague to be a collaborator and write a paper with them. And the surgeons would be happy to give tissue because even if you're not a researcher, you still like to have a paper here and there to get promoted. So like this stuff there's incentives for everyone here. And we don't do a good job of thinking that way because it doesn't feel right. And people, oh, you should do it because you're interested or because it's the right thing to do. And that's true. But especially with limited resources, you have to find ways to grow the whole area together. And a large part of that is finding ways to interact and work together on something, even if it wasn't a natural fit from the beginning or a natural thought from the beginning.
Okay, so just to mention one thing, I know rheumatology, CARA, they have that where they have created a registry across all the rheumatological centers, and then you collaborate on interact with sponsors and funders.
In PGI, we have Improved Care Now, which does that. The liver people all do this. Oncology does this at a national level. Some of our groups are way ahead of this. But we don't have this we have an emerging version of this in EOE, but we don't have it for celiac, really. We don't have it for failure to thrive or other common GI issues that we kind of come together. We don't have those pools. And so, if we to support that, we'll get there. Sub-Saharan Africa and improving water quality.
She also has a lot of stuff about, so Japan has had dramatic reductions in stunting. UK also 40% to 5%. Sub-Saharan.
So absolutely, that is true. Basically what happened is somewhere around the 80s, the rate of change of stunting went down a fair amount. In the last 100 years, there's been a massive drop in stunting. A big amount of it was caloric insufficiency. And when we fixed that, it got a lot better. What made people interested in this particular phenomenon was the realization that in some areas, there was a lack of connection with caloric insufficiency and stunting rates. And India was actually a pretty big example of that. One of the ways we initially discovered this, one group was in India, one group was in Brazil. But the group that in India was noticing, why are Muslim children in India having better health outcomes until they're 8 than Hindu children? Because socioeconomically, that makes no sense. Muslim children in India are generally the poorest demographic. In every other country in the world, SES correlates pretty well with pediatric health outcomes. And in India, it doesn't. And it was very confusing. And then you look at grown Muslims in India versus grown Hindus, and the rates of metabolic disease were way lower. So that was weird too. And the thing that they came up with was essentially behavioral differences based around the hygiene rules in Islam that are not present in Hinduism. And that was the only thing they could determine. And the fact that these people, and in retrospect, I think they should have maybe noticed the higher amount of animal consumption because Hindu Indians do not eat very much animals. So it's there's gaps there. I think what we've seen as a tendency is that we stalled out on our improvements in linear growth in like the 80s and 90s, and that's where we're sort of are right now. And we've kind of hit if you're not having an active disaster or an active conflict, starvation is relatively uncommon, but micronutrients and variability in diet is also pretty bad. So there's a sort of mixed exposure that we need to figure out here. My general hope is that tryptophan by itself does enough, because sometimes you find these regulatory buttons that you can press, like GLP-1, that just have a lot of positive effects downstream biologically. And I'm hoping that if we replace that, essentially homeostasis regains function through other compensatory mechanisms that we normally respond to inflammation.
I really enjoyed that, Tom. That was a lot of fun to hear about. I had sort of back to the science stuff. One thing that we see a lot in neonatal brain injury and lung injury is this arrest of a developmental process. And I really love this concept because we see often in all of medicine that disease recapitulates development. If we kind of get things back on track, that things go better. So I'm wondering if the arrest or the de-differentiation you see is actually an arrest of normal differentiation because these patients are exposed to the same environment from birth.
There is definitely part of that. What the de-differentiation, you know, is de-differentiation because you can still see markers of the other cell type that was further down the differentiation pathway. So we can say that we're still displaying some markers of being an enterocyte. We're still displaying some markers of being a goblet cell. We're displaying some markers of being an EEC. So that could reflect a partial shift that stopped in the middle, but that would be not something we've seen as much of. The thing that we generally observe in the mouse models and in organoid models is that you actually fully differentiate and then it changes back. Okay. Now, I do think what you're saying is accurate, and I think that is reflected in EED and part of it. Part of what I didn't get into in this is the first 1,000 days of life are really integral in stunting. If you fix stunting before the first 1,000 days are up, you're pretty good. You don't fix it, you get these long-term sequelae. And there's another point around puberty where you can make that happen again. But if you the first 7 days, what that suggests there's probably some sort of epigenetic shift. And the other thing is we can also see some heritability of some of these features. The immune tolerance effects last about 2 generations. And you can see this, for example, in IBD. If you look at IBD in the Hispanic community, IBD in the Hispanic community in South America and Central America is heavily UC. That's the way it is everywhere else in the world. If you are in the low-income nation, it's UC, if it exists at all. For example, there's 50 IBD cases in Zambia known, 48 UC, 2 Crohn's. The 2 Crohn's are both VEO IBD. And so you see this phenomenon, but if you look at a change over time, if you look at first generation Hispanic people in the United States, they have a shift where it gets about 50-50. And you look at second generation and it's predominantly Crohn's, although not as much as it is in like people who have long-term nativity. And you get to the third generation, it's completely the same. Everyone's looks the same. With some minor variations based on genetic prevalence, for the most part, it's about the same. And so this pattern recapitulates itself, and there's clearly some sort of thing that's happening there, and that is about the epigenetic set point. And I think you're seeing a lot of that too in babies, is that you have this injury that happens early on, and it kind of gets ossified, and it can't heal, even though, as we know, kids are really plastic and should generally be able to recover better because it's got an adaptive response that just gets stuck. And I do think that is a common biological tendency.
The thing that's sticking it, you see that there's a reduction in proliferation. Do you see that there's an increase in senescence? Is there a break that's on?
Not necessarily, but intestinal cells are designed to jump off a cliff so quickly that I think it's like markers of senescence are not as obvious for me, at least. I don't know if Jim has other thoughts.
Yeah, I mean, there's sort of the +4 when you get outside, like at the top of the cell niche. Those cells tend to be a little bit more quiescent. But yeah, it sounds right. These cells are normally just so short-lived.
Yeah. We don't know a great senescence signature, I don't believe, for intestinal epithelial cells because they're essentially designed to replace every few days.
And just last wonky question. How did you get to tryptophan? I just missed that.
So basically, when we looked at KEGG and went through different pathways, the gene ontology pointed towards animal protein deficiency. So then we did some network analysis and found that it looked like it was tryptophan and kynurenine as the major drivers. But it was basically through what I do in this project and what I do when I do sequencing is I'm the one who reads through the differential genes and tries to figure out patterns. Because then you can use that to inform trying to find genes that maybe weren't as differentially expressed, but might be really key in network analysis. And so I help kind of drive what genes, some of the network analysis is just looking at patterns and finding the most prevalent, but some might be saying, okay, like this is a signal, is this significant? Okay, it's significant enough. That could be a driver. And then saying, okay, let's look at what this could be involved with downstream. And tryptophan was the strongest by far. When we look at clinical data, tryptophan is also the strongest by far. Replacement of essential amino acids broadly provides a nice benefit. Replacement of tryptophan provides the same benefit. So I do think it is mostly tryptophan. It's also one of the biggest deficiencies in most of the low-income nations because it is found in sources that are just commonly not eaten.
I'm curious about it because it's like your Go terms or whatever identified, not maybe an inability to absorb tryptophan.
That's possible.
Metabolize it.
Yeah.
in which case, like maybe not only supplementing, but improving the bioavailability or improving the transport or something.
No, I mean, that's definitely possible that there's an element of inability to absorb routinely or other things like that. What we see is that the intake does correlate, and that's part of why we did the secondary work with the epidemiology. And part of what I tried to do with this talk is point out how all these things are important together, because I feel much more confident, in my opinion, that tryptophan is one of the major drivers or major risk factors for this condition. Now, because I've got, again, 3 different arrows pointing all towards the same thing in very different ways. So even though I haven't shown it causally, I definitely still haven't. To do that, I'm going to need to do an RCT. I feel pretty confident that RCT is justified, and it helps me go to someone and say, hey, maybe you should pay for this.
I don't think we need to make it. We're over time, and this is great. But I don't think we need to. Do we need to vacate? Like, no, so people are free to continue to chat. I can't stand it. I have to chat further. In our patients with eating disorders, they have malnutrition. It's basically devoid of any kind of infection.
Yep.
And we do see gut dysfunction. What we've been studying is microbial composition and function. And you know, I was so excited to talk about it, but I just want to say we are doing clinical trials, so we're looking at gut restructuring as they're in hospital, as they're being fed. We see a shift in diversity, and we're seeing some interesting things about the strains that are producers.
Yeah.
But where we're going with this in patients in the U.S. who have acute protein diet,
Yep, intestinal atrophy.
is we're going toward looked at fiber in your epi analyses.
Fiber is generally not a major issue, primarily just because of certain major sources of things like corn is major, and they tend to use the entire kernel and other stuff like that. We didn't see big gaps in fiber. Other thing that I also wonder about, like, it definitely makes sense short chain fatty acids are beneficial. The problem I would see is this essentially gas production and motility abnormalities in severely ill people are challenging. And so you might create some secondary issues and increase translocation by essentially increasing interluminal pressure. What I will say is intestinal atrophy signals look somewhat similar to this. And again, that sort of leads me to my unified theory of, I think serotonin is involved in all of this. And that there are definitely, you know, chemosensory triggers from the intestine, much of which I derive from this guy's work, or my theory derives from this guy's work, that could be linking to that effect.
Yeah, I mean, it might be a good convenient sample because there's no reason for us to scope most of our kids, but
and you know, I think that there's some interesting like meta work to do to look at like incidence of translocation or presumed translocation in people who happen to be on an SSRI and remain on it, because that would be pretty interesting, but I don't have enough of a data set for that. But I'd happy to talk about it or ways to kind of structure this in ways that my data might speak to it. Yeah.
All right, thanks. Going back to your orientation, EOB oral. Think about Ripple, think about also changing. But then you talked about you're actually looking for differences, and so maybe having a control that's closer to the pathology could actually benefit you in terms of narrowing down those differences. Did you have any celiac patients that were looking at?
No, we did actually do exactly that you're talking about there and use an intermediate case. It was South African patients. South African diets are much more full of animal protein. And so they had much more intermediate phase ED. But even when you looked at the people with more severe, there was still an animal protein deficiency signature, but not nearly as strong as it was in the Zambia. And so it suggested again that from the reason why ED is not as bad there is there. What you say is accurate. Whipples do not mean that your duodenum is intact. And that's part of why we use that and the EOE. My concern was that there might be duodenal expression changes associated with having EGID, even if you don't have obvious eosinophilic duodenal disease, or so I wanted to have another set for validation purposes, just to kind of, again, with sequencing, One thing I didn't show here, but I often do show, is the one of the, like a video of those sculptures where if you change the angle you look at it, you change what it is. Sequencing is really vulnerable to that. And so having a good comparison and having another comparison helps you make sure that some of the changes you're seeing are actually conserved and not just artifacts of what you've done. And so that's part of why you do so many analyses. That's part of why reviewers ask for validation. It's part of why you do this. And it's not unfair. Like, it is helpful. You have to look at it from every possible angle. And if you see the same picture from every angle, then you can say this is an elephant. And it's not just the three blind guys describing different things in different ways. But yeah, but no, it's a good point. Thank you, guys.
Thank you. Great talk.